Hi, I'm Robert Kale from David Rubin's lab at UC Berkeley, and today I'm going to tell you about our recent JCB paper, Induced Nanoscale Curvature Bypasses the Essential Endocytic Function of Clathrin. So clathrin-mediated endocytosis, or CME, is this complex membrane evolution from flat plasma membrane to spherical vesicle. And a lot of the molecular players of this process are already known. So AP2 initiates the process by binding to a receptor and clathrin. The site is stabilized by the FCHO1 and 2 complex, and as it invaginates into the cell body, it incorporates more receptors and cargos and coat proteins until finally it undergoes scission catalyzed by the GTPase dynamin with an assistive force produced by the actin cytoskeleton. And clathrin has long thought to be important in curvature generation, but it shows up in a lot of different shapes within the cell, visible in these SEM images. So you can see it as these spherical, flat, or tubular lattices. So how and when curvature is generated during CME has been a topic of debate for some time. So in order to study curvature generation during CME, we developed this nano-ridge substrate system, which you can see in this top-down SEM. There are these thin ridges with thicknesses from 75 up to 1,000 nanometers. And then when we grow cells on them, visible in this thin section TEM, the cells respond to the ridge by forming this semicircular shape as a function of the nano-ridge size. So this allows us to induce curvature on the ventral cell membrane, sort of separated from cell intrinsic processes like CME. And then when we image cells growing on these nanoridges on a light microscope, you can see that the entire ventral cell membrane is visible, sort of evenly distributed across the ventral surface. But when we image endocytic proteins like AP2, RFP, or dynamin 2 GFP, there's this really striking enrichment of the endocytic proteins, specifically on the nanoridge substrate sections, with a more modest increase as the ridge diameter increases. And when we quantify this, we can actually see that there's a 3 to 4-fold enrichment of endocytic proteins like the early proteins AP2 and clathrin light chain A, and a greater than 4-fold enrichment of the intermediate or late scission proteins, epsin1 or dynamin 2. Meanwhile, caveolae, as marked by cav1 RFP, are essentially curvature insensitive. So there's something specific to CME that's localizing it to these nanoridge structures. So we next wondered if we could perturb some part of CME that would cause it to fail on flat substrate, but be rescued with induced curvature. So for instance, when we knock down the stabilizing proteins FCHO1 and 2, we still see really robust enrichment of AP2 and dynamin 2 on the nanoridge substrates. But these sites abort rather than committing to endocytosis. So this tells us that nucleation itself preferentially occurs at the nanoridge substrates. Meanwhile, if we knock down clathrin, there is no change in the enrichment of AP2 or dynamin 2, so their localization to curvature is independent of the clathrin coat. And there's also a really strong rescue of overlap between the two. So when you knock down clathrin, you see a decrease in overlap on flat substrate, but you see no change on the induced curvature. So this tells us that the sites are forming sort of normal endocytic structures. And additionally, when you follow these sites over time, you can see that on flat substrate with clathrin knockdown, AP2 is weak and dynamin 2 recruitment is sort of noisy, indicating that these are abortive sites. Whereas when we image these same cells on the nanoridges, we see a really strong increase in AP2 fluorescence and a sharp late burst of dynamin 2, which is sort of canonical of productive vesicle formation. So we wondered if these sites could actually take up cargo. And as you can see here in these XZ projections from confocal microscopy images, in control cells, when you expose them to fluorescent transferrin, they take up very many puncta fluorescent transferrin, and these form these perinuclear puncta, which is evidence of being trafficked from the cell surface. But when you knock down clathrin, you see sort of weak membrane-bound transferrin puncta that are on the periphery of the cell rather than trafficked near the nucleus. But with these same cells on induced curvature, we actually see an increase in fluorescence of transferrin and a rescue of the perinuclear localization. So this is evidence that these vesicles actually form perfectly normally with induced curvature, even absent the clathrin coat, which tells us that clathrin's role is to stabilize the induced curvature. So in summary, enhanced nucleation occurs at curved membranes. And also when clathrin is knocked down on flat sites, there is disassembly of the endocytic site, whereas with stable curvature, the other proteins are sufficient to form an endocytic vesicle, and therefore clathrin's role is to stabilize induced curvature. Thank you.